Hello, I am Grace Lai, a neurosurgery resident working with Dr. James Drake at the Hospital for Sick Children. Today I will be presenting on an in vivo porcine model of intraventricular clot lysis using MR-guided high-intensity focused ultrasound. Intraventricular hemorrhage is a major risk for premature infants. An area of the brain called the germinal matrix, when not fully formed, has fragile blood vessels that can easily bleed. If there is enough blood, the fluid spaces, or ventricles, in the brain can get clogged, which results in buildup of cerebrospinal fluid. This condition is called hydrocephalus and can be fatal if not treated surgically. For many years, neurosurgical interventions all involve removal of excess spinal fluid. However, there have been recent efforts to reduce the risk of forming hydrocephalus through direct removal of intraventricular blood clot through an endoscope. At Sick Kids, we are developing technology for incisionless targeted hematoma lysis using MR-guided high-intensity focused ultrasound. For treatment, we are using the Profound Sonolief system modified for cavitation. Our lab has previously developed a porcine model of intraventricular hemorrhage through removal of the skull and direct injection of blood into the ventricles. After the blood is infused into the brain, the animals are treated using the Profound system. The pig is placed on its back with the top of its head resting on a gel pad. Previous studies from our group demonstrated successful clot lysis on immediate post-treatment pathology examination. Through in vitro and in vivo testing on acute animal models, we have determined that these are the parameters that have shown the optimal efficacy. The experiment is designed as follows. The animals get an MRI before and after surgery for injection of the blood clot. The next day, they are taken back to MRI for HIFU treatment. A combination of the axial, coronal, and sagittal T2 MR scans are used to place the treatment cells in the clot, taking care to ensure that the near-field end of the seed does not touch the proximal ventricular wall. We also ensure during planning that the HIFU beam does not interfere with the skull aperture. Standard MR thermometry is used during treatment. Afterwards, the animals are monitored daily for neurological deficits and get serial MRIs weekly until 28 days. This graph shows the ventricular volume over time as measured on MRI. 5 animals were treated in total. 1 animal died immediately after treatment and was not represented on the graph. Another animal, represented in red, developed worsening neurological status and was terminated on day 5. 3 animals survived until the planned endpoint. The one in blue had parenchymal damage from far-field effects and showed sustained ventricular dilatation. The one in green never developed ventriculomegaly, and the one in purple showed initial ventricular enlargement followed by resolution. We know from our experience developing the survival model that the initial amount of clot formed is proportional to the degree of sustained ventricular dilatation. Indeed, the pig in green had the smallest amount of initial clot, while the blue and purple had similar clot volumes. For the first treated piglet, we observed far-field effects for seeds where the target was located in front of the nasal sinuses. The ultrasound waves likely reflected back toward the transducer when they reached the air tissue interface distal to the target. The right image is a side view with the top of the head closer to the transducer. The sinuses are located in the area of the orange circle. While the animals survived until the end, there was lasting brain damage in the location where we observed the far-field effects. We subsequently filled the nasal cavity with ultrasound gel prior to treatment. We did not observe any further far-field effects in subsequent animals. The animal who died immediately post-treatment suffered severe frontal lobe damage from near-field effects. This pig was treated at 500 watts while the rest were treated at 400 watts. This was the only treatment where we observed near-field effects, which started after several treatment cells. However, once it started, we observed rapid onset of near-field heating on all subsequent attempts. This is likely due to air bubbles formed in the brain tissue during the first instance of proximal spread of the cavitation cloud. Due to the location of damage, the pig suffered severe neurological impairment upon waking from anesthesia and required compassionate termination. Finally, we ran into an issue where one of our piglets were not properly supported and had a shift in head position after initial localizing scans. Cavitation of the brain occurred instead of the blood clot. This highlights the importance of head immobilization when not using a stereotactic frame where the head is secured with pins. Despite mishaps faced during this pilot phase, we have learned valuable lessons moving forward. We have determined the upper limit in terms of power on the Sonolief machine. We learned that treatment must be terminated following the first instance of backpropagation of the cavitation cloud into the intervening brain parenchyma. As always, immobilization is of vital importance, and a reliable means to do so must be available with any potential frameless system. Finally, our results are promising for the 2 animals where we did not observe any adverse events. In fact, one animal showed resolution of initial ventricular dilatation. A larger, adequately powered study comparing treated versus non-treated animals will address the hypothesis that clot lysis can prevent subsequent hydrocephalus. Our lab is also actively developing cavitation monitoring and acoustic radiation force imaging to improve our ability to monitor treatment safety in real time.